All right, so in anatomy and physiology, we're now starting Unit 2. Unit 2 only has 2 chapters in it, histology and integumentary. So I'm going to do one lecture on histology, one on integumentary. All the photos for the histology, if you haven't noticed, have already been uploaded onto Canvas, both with low power objective under a microscope and a high power objective. People ask, which am I liable for? The answer is yes. That's why it's all there waiting on you. Also, the similar photos are going to be found in your lab manual with the descriptions. I'm going to go through these just so that you hear me and see me go through it with you. I'm going to walk you through the photos. I'm not going to have the photos with me. I'm going to sketch them out so you can have an idea what we're looking at. But you have the photos in front of you. So whenever you do this, again, understand there's going to be a lecture test and a lab test. The lecture test is going to be on the vocabulary, on the traits. It's more the physiology of how it works. The anatomy part is this is this is that, this is it. Basically, you're pointing out, the features that are there. In your lab manual, I don't think I brought the lab manual up with me to my office. I didn't. But you'll see at the end of your, in your lab manual, it's it's one of the last pages of Unit 2. It'll have a pick list, which is basically what you're going to use for your lab practical. So if you know those features on that page, I forget how many there are on that page, but there's letter designations like AA means this, A is this, A, whatever. But that's all on that page, and as long as you know those features, you know what you need to know for your lab practical for histology as well as integumentary. But those are the only questions I can ask you. Those can be the only answers. Okay. Now for the lecture part, that would be more like how does this work? Where is it found in the human body? So on and so forth. Okay. So here we go. Histology. So histology means the study of tissues. You're going to be careful because whenever you did your review, the science of cytology is the study of the inner workings of a single cell. When you have groups of cells doing similar functions, that's obviously going to be then a tissue, which is histology. So an organ. An organ, you should know the definition of. It's when we have 2 or more different tissue types and they're doing similar functions together. But there has to be 2 or more tissue types, and there has to be a discrete boundary, meaning a definite beginning and a definite ending. You can't just blend one into the next. Next you see there the primary germ layer source, the stem cells that will lead to this. You are considered to be what's called a triploblastic individual. When you were developing inside mommy, you had 3 germ layers, meaning ectoderm, mesoderm, and endoderm. Your ectoderm is what's going to eventually create your epidermis, as well as your central nervous system, your brain, as well as your spinal cord. Once we go to mesoderm, it's a lot more involved than just what does it make, because it's easier to talk about what it doesn't make than what it makes, because it makes pretty much the majority of you. Everything from your skeletal system, muscular system, cardiovascular, reproductive, the list goes on and on and on and on. All right, endoderm. Endoderm means it makes the inner lining, not the actual digestive tract, but the inner lining of the digestive tract, the GI tract, the respiratory tree, and organs that would support digestion. Things like the liver, gallbladder, things like the pancreas. That would come from the endoderm. Anything that I didn't mention would be mesoderm. All right, now. There are 4 major types of tissues. You have epithelial, connective, muscle, and nervous. You have to be able to differentiate between the 3. Epithelial is going to be the most superficial of all the layers. It's avascular, meaning no blood vessels go directly toward it. Since there are no blood vessels going directly toward it, it means that its availability of energy is very low, which means its cell cycle is normally quite slow. Unless it becomes injured, then it can ramp up production, but once it's healed, it will ramp back down to normal levels of ATP usage. Epithelial means also too that these cells are going to have 2 surfaces. The upper surface, the superficial surface, is going to be called the free surface, meaning it's not attached to anything else. It's the apical or free surface. The other side, which is touched to everything else below it, that's going to be the basal surface or the basal membrane, like this. So if I have cells, let's say up here is the outside world. There's my cell, okay? This surface doesn't touch anybody else. Okay, and let's just say there's tissue down here, all right? This side doesn't touch anybody else. This is called the apical surface or the free surface. This surface down here that touches other things is called the basement surface, the basal membrane, or the basement membrane or basal surface. Okay, that's the only those are the only time you see them is with epithelial tissue. Where do you see epithelial tissue? Again, on the outermost areas of your body. So what's going to be its function? Well, everything from protection, absorption from the outside, secretion from the excretion. All these are functions which are going to be through that interface between one barrier and another. All right, connective tissue. Connective tissue is the largest of all the four categories. It's usually the most difficult, not because it's hard, but because there's so much underneath it. Connective tissue is going to have underneath it 5 subgroups, which are going to be loose connective, dense connective, cartilage, osseous, and blood. They all fall into that category. So if you look at the types of tissues that are in there, you have adipose tissue. So functions of connective tissue could be everything from, if it's adipose tissue, energy storage. If you just do the bodies of organs, it's often called the soma of an organ, the bodies. The soma is Latin for body, so soma of an organ. Any sort of squishy organ, think of like a spleen or a liver, that's going to be connective tissue, so it makes up the body of it. If we look at cartilage, cartilaginous tissue is meant for support. It's also meant for lubrication. It's also meant for a buffer between bones. If you then also look at osseous tissue, bone, well, that provides us a structural component for your body, a framework. It also is a repository for minerals and mineral homeostasis. So lots of functions going on with that. Lastly, we have, blood. Blood doesn't connect anything, but because it's derived from connective tissue, it's classified under connective tissue. So therefore, blood comes from bone marrow, which is myeloid tissue, which is connective tissue. From there, you get all components. You get erythrocytes, leukocytes, platelets. The list goes on and on. The ones you're liable for are going to be, leukocyte, erythrocyte. All right. Then we go to muscle tissue. Muscle tissue and nervous tissue, those two together are what we call excitable tissues, meaning they have the ability to initiate an action potential or a nerve impulse or propagate it through the tissue. Muscle tissues propagate through the tissue. Because of that, that means that they have to have some sort of stimulus in order to activate. That's why they're excitable. Well, there are 3 main groups of muscle tissue. We're going to have cardiac, smooth, and skeletal. All 3 involve with one major function, motion of one sort or another. Skeletal means attached to your skeleton, which means, gross motor movement, raise your arm, wink your eye, that type of thing, turn your head. Cardiac, obviously, is for cardiovascular. Smooth muscles scattered around your body. It's like the muscular walls of the uterus. You see it in walls of the walls of the gastrointestinal tract for peristalsis. Basically, your intestinal tract acts like a tube of toothpaste that you squish the contents through. As you contract it, it squishes the contents throughout. You also have in your piloerector muscles, which are what give you goosebumps. So it's scattered around your body. Nervous tissue. Nervous tissue is also considered to be what's called excitable tissue. Again, this is what's going to initiate the action potential. Nervous tissue is all about communication. It's all about coordination of body activities and also bringing in external stimuli and coordinating those activities. All right, so those are the 4 general types. The rest of this is going to be going over each of the 4 general types and give you more specifics on them so we can compare and contrast between them. So, first one, epithelial. Epithelial, I already told you, has major traits. Number 1, it's avascular. No blood vessels leading directly toward it, which means it's going to be number 2, energy poor, meaning that its cell cycle cannot be very quick because it can't do cell division without energy. Also, it takes a while for waste to come out. How does energy come to it, nutrients, and how does waste leave? It's through diffusion. It's a slow process. So therefore, not the fastest cell cycle, but again, they can regenerate quicker if they become injured or impeded upon. They are the only ones which are going to have an apex, apical surface, or a free surface, and a basement membrane or a basal surface. Along with that, when we talk about tissues, how we usually put them into categories is based upon 3 major characteristics. Big star. The 3 characteristics are cell type. Second one, is going to be what's called the ground substance. The ground substance is the material surrounding the cell, whether it be hard, gelatinous, liquid, whatever it is. And then number 3 would be the proteins that connect in between. Now, number 2 and number 3, The ground substance and the protein fibers together collectively are called the matrix. So when you see in your lab manual the matrix, that's the ground substance, and then also then the protein fibers that connect. The cell types are on their own. Now when we get to epithelial is the only one we're going to talk about that's not going to have a matrix. Why? Because the cells are packed close together, and therefore there is no surrounding tissue. Basically, think of like bricks in a wall or cobblestone on a street. They're basically stacked side by side, and therefore there isn't a matrix. The other ones will have a matrix. They'll have an extracellular matrix, ground substance, and they'll also have the protein fibers. All right, moving on. How are we going to differentiate between them? Big start. You differentiate epithelial tissue based upon, number one, the number of layers that compose it, and number two, the shape of the cells that compose it. Okay? So, if I only have one layer of cells, that is called simple. If I have more than one layers of epithelial cells, that is going to be then called stratified. If I have one layer of cells, but it looks like it's more than one layer of cells, that is called pseudostratified. So here we go. Let's just do a simple one. That would be called a simple layer. Now, if I had another layer below it, now that'd be called stratified. Now, how I know stratified from pseudostratified, if you notice there's a definitive barrier between them and I can see my nuclei are in these different rows. Pseudostratified means you usually have these long skinny cells, but they're not geometric as you would think. They kind of worm around, and therefore they're not specifically any major shape, and therefore it tricks your eye. Notice how when I draw these things, the nuclei tend to be like all little ducks in a row, all lined up. In pseudostratified, even though it's one layer, the nuclei are going to be juxtaposed all around different areas. So when you look at it, you go, oh, look, there's different, I can see different rows of nuclei. It's more than one. Look closer. There's only one apex, one basement. It's one long, skinny, weird-shaped cell, but only one cell. So it looks like it's more than one layer, but it's only one layer. Pseudostratified. Okay? Now, the other thing you do to classify epithelial tissue is the cell shape. If it's square, that is called cuboidal. Now notice, cuboidal again, you'd think it's not two-dimensional, it's three-dimensional, it's a cube. But the thing you notice is the nucleus is smack dab in the middle. If I only had one layer, that would be called simple cuboidal. If I had two layers, that would be stratified cuboidal. Okay? Now, if I have, instead of squares, I have rectangles, that would be called columnar. But notice when I do this, my nuclei are going to be toward the basement membrane. They're away from the apex. So when you see nuclei that are rectangular with the nucleus toward the basement membrane, that would be stratified. Now again, that would be simple stratified that I just drew. All right. Now another one is called squamous. Squamous means flattened. Flattened means there's no particular shape. Basically, take a ball of clay and just squish it. Every time you do it, you're going to get a different shape. That's squamous. So basically, squamous can look just like really just squished cells. Just squished. Okay? Now, more than likely, in Gen Bio, when you had Gen Bio, when you did the microscope, you did a cheek scrape, and you looked at it under the microscope. What you were looking at were stratified squamous cells. When you look at them, you notice none of them were the same shape. Right. They're just flattened, weird. It kind of looks like little tiny clear sandwich bags that have deflated, so they look like raisin sandwich bags is what they look like. But they're all different shapes. The other one then, of course, you have flattened, squamous. You have then square, which is cuboidal. You have rectangular, that's going to be columnar. Those are nice, neat categories, but then you also have to have your group other. Other in this case is going to be called transitional. Transitional means that as the tissue stretches, the cell will change shape. That is going to be called distension. When you stretch the cell out, you're distending the tissue. And therefore, it'll change shape as it does it. And that's called transitional. Now, one of the hardest parts people have with histology, specifically epithelial. What if I had layers of tissue, epithelial tissue, my top layer, my most superficial layer, was columnar. Then let's just say for S and G's that my next layer down was cuboidal. And then let's just say I had squamous. You know, I've had all different, it's never going to happen. Let's just say I do that. We could all agree there's more than one layer of epithelial tissue, so you know it's stratified. But what shape do you call it? Big, huge star. You always label it with the shape of the cells which are found the most superficially. This would be called stratified columnar. Why? It's more than one layer of epithelial tissue cells, but the most superficial layer are columnar. So it'd be called stratified columnar. Remember that. Now, the rest of this you see in my outline, I've already written out for you, okay? So basically all I'd like you to do is when I put simple squamous epithelium, basically you read my description of it, the functions of it, where it's found in the body, and then you look at the picture associated with it. So basically when you see the picture, by the time when you do this, like a flashcard, if you saw the picture, you should be able to go, that's simple squamous, it's found blah, 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 blah. Basically, if you flip the card over, that's what you would write on your flashcard. That's the level you need to be at for this exam. Okay, so I don't need to read what you already got written out for you, but I'll summarize. Okay, so simple squamous epithelium. Think simple, one layer, squamous, flattened cells. These cells are going to be in areas where you're going to need things like areas of very little wear and tear. If you remember back in Unit 1, I talked about what are called serral membranes. That was one lecture I gave about the terminology. That's when I talked about visceral and parietal, the outside surface of the lung, then the inside surface of the rib cage. Those would be simple squamous epithelium. So therefore, they tend to give off a lubricating fluid. They have a very, very slow regeneration time, so if injured, they take a long time to repair. When you look at them, they kind of look like almost like a spider webbing under a microscope. Think of like almost like a cheesecloth almost. That's how they'll look like, but you'll see that in the picture. Simple cuboidal. Simple meaning one, cuboidal, square cells, nucleus in the middle. Great. Where do you find these? Areas where you have secretion, giving off things. Mostly you think about like in glands where you're giving off hormones, like your adrenal gland. And then you also see things like for absorption, bringing things in. But mostly you think about secretion, like for gland tissue. Now follow, this is nature, okay, you understand? So when I say simple cuboidal, don't think like a hard square. Take the square and knock the edges off. So I can have a ring of squares like this. That would be simple cuboidal. Why? This would be the free surface. Therefore, these cells would secrete their hormone, put them into this little tube, and then it would go out to the rest of your body. Okay? Then you'd have tissue surrounding it, and you might have another one of these to give off the same hormone. Okay? Now, that's simple cuboidal. Next, then, you have simple columnar. Simple columnar, again, means one layer of rectangular cells, the nucleus toward the basement membrane. These are going to be in areas where you're going to have like absorption. So you can have simple columnar that are just simple columnar. I put that picture in there for you. You can have simple columnar which are ciliated, have little hairs on them to help move things across the surface. If you do have that though, you can have another feature called a goblet cell. Goblet cells, big star, are cells which secrete mucus. So let's just say I had this. Long skinny cells, and then every so often I'm going to have this like void in the middle of it. That would be simple columnar, one layer of rectangular cells. These cells that look empty are going to be secreting mucus out to the outside free surface. These are your goblet cells. In a lot of cases, what you'll also have too, you could either secrete the mucus with no cilia. If you have cilia, what's that doing? It's moving the mucus back and forth. It can use to trap pathogens, allergens, whatever, to get it out of there. Now, if we look at the inside lining of your small intestines, your gut, you would have, not ciliated, they would have microvilli. At the free surface, what you would see are these little tiny finger-like projections coming off the surface of the small intestines. Now understand, on a slide, when it's made, when the microtometer comes down, all this is like soft bread and it gets squished, so you're not going to be able to see that on a slide because it's what's called an artifact. But these are called microvilli. They're not cilia. Cilia look like little tiny protein hairs. They even when you cut them, you still keep the integrity of the hair. These get squished like fresh bread. What do? They're increasing the absorptive area to absorb nutrients, in this case from your small intestines into your bloodstream. Okay, that's all they're there for. But however, you understand your small intestines also has mucus lining on the inside of it. Why? To get rid of pathogens, basically, is all what it's there for. Okay? So now, understand, you can have simple columnar without goblet cells. But if you see goblet cells, they have to be columnar cells. They got to be columnar epithelium. All right, continuing on. We then have simple columnar ciliated, which I already talked about. If I have cilia on the surface, where would you think you'd see that? Every time you go, well, that's the mucus, and that's the cilia trying to get it out. That's the mucociliary escalator. Now, stratified squamous. Stratified squamous means more than one layer of squamous cells. Okay? So, stratified squamous, you're going to see 3 ways that I put on canvas for you. First way is a smear. This is basically when you do cheek smear on the inside and you spread the cheeks out. So basically, stratified squamous just looks like a bunch of cells, scattered around an area. How can you tell they're stratified? You can't. That's why it's a smear. You can tell they're squamous because they look like little squished raisins is what they're going to look like. But you have to remember those are stratified. Now, keep in mind, stratified squamous is going to come in 2 types, keratinized and non-keratinized. Keratin is a protein which waterproofs your skin. Well, anything on the outside world, that's keratinized. However, if I go the inside of your mouth, your esophagus, if I go up into the vaginal orifice, those are non-keratinized stratified squamous epithelial cells. So therefore, they always have to be kept damp because otherwise they'll dry out and they'll crack and they'll bleed and invite infection. All right, so your cheek smear obviously would then be non-keratinized on the inside of your mouth. Now, another slide you're going to see then is going to be a whole mount of skin. That would be like you basically looking at your skin, looking straight down on it. So what you would see are, a bunch of cells like this, each individual nuclei. It kind of almost looks like chicken skin. But then every so often you see this big dark glob somewhere in it. Okay? Understand what you're doing is you're looking down at your skin. What are the dark globs? That's melanin. Melanized patches of skin. So in this case, you're not seeing true stratified. You're looking at the whole surface of your skin is what you're doing. If you want to see the actual true stratified squamous, look at the next slide. It's going to have a cross-section of your skin. Your integumentary system looks like this. Okay? This upper surface up here, that is your epidermis. This area down, there'll be large bubbles down here. This area here, from here to here, that is your dermis. And down here, that is your subcutaneous, also known as the superficial fascia, also known as the hypodermis. Has 3 different names. Okay? All this down here is the fat tissue of your skin. This is your dermal layer. This is your epidermal layer. Stratified squamous exists in the epidermis of your skin. In this case, if it's facing the outside world, it would be keratinized. So what you would see are little cells all along the boundary, and then as you go up, they get smaller and squished kind of flat. Well, think, why? These are avascular. The blood's down here. The further you get from this boundary, the further you go toward the outside, you have less and less nutrients. What are the cells doing? They're starving. And as they starve, they die and eventually gel together to become what you look at. And when you look in the mirror, your skin, your stratum corneum. Okay? But that would be stratified squamous as well, too. In that case, then it's a protective covering. Stratified cuboidal means more than one layer of cube cells. You would see this mostly if you went to the next section in your in Unit 2, and you went to skin. If you look at the eccrine glands, your eccrine sudiferous glands. Sudiferous, S-U-D. Sudiferous means sweat gland. You have to know the proper name, okay? So eccrine sudiferous glands are the glands that you use when you actually sweat. The gland itself has a terminal duct on your surface, so it basically floods the surface with water, urea, lactic acid type of stuff. But the sudiferous glands, when they're down here, they kind of look like these large, huge, looks like tire tread. Looks like this. And then you have the duct come to the surface, okay? But if you look at these tire tread, there'll be 2 layers of cuboid cells around each circle. That would be stratified cuboidal. Stratified columnar. Stratified columnar, again, would be the one I drew earlier where you'd have more than 1 layer of epithelium, but the most superficial one would be columnar. Let's see, transitional. Transitional epithelium is going to be areas where you're going to have distension. Distension, if you remember, means stretching. Anything dealing with your excretory system. Think your kidneys, the tube coming from your kidneys down to your bladder, that's called the ureter. Then you have the inside lining of your bladder, and then you have the tube from your bladder leading out the urethra. All that is going to be transitional epithelium. When you see transitional epithelium, it kind of looks like this scalloped, wavy layer, like that. That would be transitional epithelium all along here. Understand why it's wavy. It's because it hasn't stretched yet. Whenever you have to go pee-pee, you're doing the pee-pee dance, you feel the pressure, that's when this gets stretched out, and therefore causing pressure, feeling that you need to, urinate. So therefore, but at this time, they may be at this point, they may all look, combination of cuboidal and stratified. When it stretches, they may look squamous. So the cells change shape as the tissue stretches. That would be transitional. Pseudostratified columnar epithelium. This is the one where it looks like it's more than 1 layer, but it's not. It's only 1 layer, and that was the one I showed you before, where now the cells are all these weird little shapes, but the thing that throws you off are that the nuclei are not all neatly in a row, so you think it's more than 1, but it's not based upon the placement of the nuclei. These can also have goblet cells that come out. These are the ones that also could be ciliated as well too, and therefore they can move the mucus along the area to clean an area out. Pseudostratified columnar epithelium you can find in areas like your upper respiratory tract. That's going to be pseudostratified as well too. Okay, so much for epithelium. Now we go to connective tissue. I'm going to leave this picture up because we're going to refer to it as we go further along, okay? So remember, epithelial, stratified, squamous, all right? So next one is connective. Connective, if you remember, is going to have all 3 features. It's going to have cell types, it's going to have protein fibers, and it's also going to have the ground substance surrounding it. Now, connective tissue is going to be vascularized. Lots of blood vessels, lots of nerve endings. It has all little bells and whistles all throughout it. However, unlike epithelial, there is no free surface. You're connected in every which area. There is nothing that's unattached, that's apical or free. All right? Now, connective tissue. Depending upon what it is, depends upon how vascular it is. Now, keep in mind, when you're when you're inside mommy as a fetus, your cartilage is going to have blood vessels in it. It's also going to have nerve endings in it. However, as you mature and it becomes adult cartilage, also if you think about your tendons and your ligaments, what's a tendon? Tendon is connective tissue that connects a muscle to a bone. A ligament is connective tissue that connects bone to bone. That's going to be avascular as well. Your cartilage is avascular as an adult. When it's bone-forming cartilage, when you're a fetus, well, yeah, there's blood to it. But in you right now, your cartilage is avascular. Also doesn't have any nerve endings in it. If you ever heard the old saying, don't cut your nose off to spite your face. Well, why? This right here is hyaline cartilage. If you were to have this cut off, it'll never grow back. Why? Because the cartilage has inability to grow back. If you have somebody who's an athlete, soccer, football, track, and they blow out the menisci in their knee, the menisci is fibrocartilage. If they blow that out, it doesn't grow back. Basically, you get arthroscopic surgery, try to knit it back together again and hope that it stays. What you become is a poster child for an artificial knee later on in life. So therefore, cartilage, because it doesn't have blood vessels, cannot grow back. Same thing for a tendon and a ligament. Tendon and ligaments are avascular. So if you tear a tendon and you tear a ligament, they basically have to be knitted back together again artificially. All right, so moving on. When we talk about connective tissue, talk about tissue types. Whenever you hear the root word cyte and blast, you got to be careful. Cyte means an adult cell. Adult cells are all about maintenance, which means they need less energy. Blast means immature or forming, meaning they need lots of energy and therefore they're not the full mature cell yet. So when I go through this, if I talk about chondroblast, Quandro is the word Latin word for cartilage. Blast, immature cartilage cell. Chondrocyte, adult cartilage cell. Osteoblast, immature bone cell. Osteocyte, mature bone cell. All right, so other cell types you would see. One of the major things you're going to see there, big star, are going to be what are called fibroblasts or fibrocytes. People miss this on a test, and I want to throw something at them. Fibrocyte. I am the cell that makes the fibers, the protein fibers. So the fibers I'm talking about, things like collagen, elastin, those are going to be made by these cells. Simple enough. Let's see, moving on down. Mast cells, put a star next to. You found these in blood vessels. Mast cells are going to create histamine, which hopefully you know is an anticoagulant. Therefore, your blood doesn't clot in the major vessels. Also, too, it's going to create. Histamine is for swelling, inducing edema. I was thinking heparin, but mast cells also create heparin. Heparin. Heparin is the anticoagulant. Histamine promotes edema, promotes swelling, retention of water, retention of the cells. That's one of the first immunological responses you get. So therefore, as the cells swell, it prevents things from getting between the cells' tissues in the ground substance to move on further in the body. It's kind of like putting police tape around an area, cordoning it off. All right, adipocytes, big star. You should know those mean fat cells. And also in your lab manual, before I forget, there's a page that talks about the different types of connective tissues. And it tells you this is the name of the connective tissue, these are the cell types, here is the ground substance, here are the protein fibers. Offhand, again, I didn't bring it with me. It's one page, it's a data table. Help yourself out, start memorizing it as soon as possible. That's the best thing that's going to help you. It's the easiest way to do it. Now, if you'll notice, one of the most common ground substances is going to be something called hyaluronic acid. Hyaluronic acid is a substance which basically is viscous but slippery. So think of it almost like a hair gel, but it's like a silicone lubricant as well. Where would you find hyaluronic acid? Well, it's in your skin. You have it in your joints, therefore it makes a lubricant so your joints work better. Hyaline cartilage makes hyaluronic acid, which I'll talk about later. But it's more, basically when you talk about plain connective tissues, it's all over the place. Okay. When you talk about adipose tissue, fat cells, its ground substance will be triglycerides. That's how your body stores fat. If we talk about cartilage cells, anytime you hear the word chondroitin sulfate, chondro, cartilage, that is the ground substance of cartilage cells. For bone, osseous tissue, anytime you hear the word calcium, calcium phosphate, calcium carbonate, calcium salts, that is the ground substance. It makes it hard. For blood, the ground substance is real simple. It's plasma. Plasma is what holds everything in it. All right, so just memorize those. All right. Moving on down, then we talked about the cell types, talked about the substances, fibers. Again, there are three major protein fibers: collagenous, elastin, as well as reticular. The two you have to be able to distinguish on a slide would be collagenous and elastic. So here we go. Collagen, when it's alive, is a white fiber. If you ever saw somebody or you have stretch marks, those are collagenous fibers. They're called striae. What happens? In your dermis down here, you're going to have collagen fibers everywhere. When you take the skin, you stretch it a lot, the skin does not want to rip. So what happens? The collagen fibers tear, and then they reform as quickly as possible, usually in parallel lines directly associated with how it was stretched. The whole idea is to prevent it from ripping. So when you're looking at somebody's stretch marks, you're looking at the collagen in their dermis through their skin. But collagen tends to be, when you see it on the microscope, they're thick fibers, almost like rope. And anytime you see collagen, just in your brain think ding ding, strength. It's there for strength. Hold this thing together. Elastic fibers are made up of the protein elastin. Elastin fibers, when it's alive, are yellow. Whenever you see them on a microscope slide, they tend to be little thin hairs, and they tend to be stained really, really dark, like navy blue, almost purple. So it looks like a real thin thread going over it. Well, what are they for? Well, they're helices. So therefore, the skin that area can stretch, but like a spring, go back. And that's what they're there for. Reticular fibers are a special kind of collagen where you have glycoprotein attached to it. So it looks like this rope-like cord that looks furry. You don't have to recognize that on a slide because it is difficult to tell. But basically, reticular fibers are anywhere where you need support of a body of an organ. Like I was saying, whenever you have organs, when you think about that are squishy, like the liver, like the spleen, they're squishy. Those are going to be, what makes up the body of it is going to be reticular fibers. All right, so here we go. There are 5 subclasses of connective tissue. We got loose connective, we got dense connective, you got cartilaginous, osseous, which is bone, and blood. You have to be able to compare and contrast between them. All right, so here we go. Underneath loose connective, there are 3 examples that I give you: areolar, adipose, excuse me, and reticular. All right, so those are the 3. So, first one, areolar. Where do you find areolar? You can read as well as I can on the outline, but one of the major areas, come back to my drawing. If you remember up here in the epidermis, this was stratified squamous. If you notice in the dermis has this wavy area on top and this big area down below. With your imagination, make a dotted line underneath those humps. This area, that's the hump area, it's what's called the papillary region of the dermis. Papillary region of the dermis. Imagine you took plastic wrap and you took your fingers and gently pushed up and you'd make this ripple. That's what you'll be seeing. Why do I mention this? Because areolar tissue is found in the papillary region of the dermis. Okay? Now, what does areolar tissue look like? When you look at the slide, please note, you'll be able to see the collagen fibers. They look like big ropes. You'll see the blue threads, the elastin. You'll see like little tiny beans inside of there. Those are the fibroblasts that made the protein. And you'll see like what looks like a lot of empty space. Keep in mind, when the tissue was alive, that's hyaluronic acid, the ground substance. All right? Adipose tissue. Adipose tissue, to me, when I look at it, looks like fish eggs. If you think like salmon eggs, if you ever eat like sushi, if you look at the masago sitting on it, like little tiny orange balls. Now, fat, you understand, doesn't have any sort of color to it. You have white fat and brown fat. As an adult, you got white fat. It has a little yellow tint to it due to a pigment called carotene, not keratin. Keratin waterproofs. Carotene is a pigment you see in fat cells. Example. You notice at the palm of your hands kind of like a yellow tint to them. That's because there's no melanin here, and therefore you're seeing the fat pads in your skin, and therefore that's the carotene that you're looking at. So with that, it's also in the stratum corneum. It's outside of your skin. Anyway, fat cells, what are they there for? Well, think long-term storage of energy, insulation. They're also there for a shock absorber around organs as well too, so they don't get injured. What do they look like again when they're not stained with Sudan 4? They look like soap bubbles. Imagine you took a bath and you have these large soap bubbles. That's what they look like. What's mostly composed of? Triglycerides. That's how your body stores that long-term energy. If you stain it with Sudan 4, those soap bubbles then tend to look hot pink, or if they've been sitting around long enough, they kind of look reddish orangey. They're large bubbles is what they look like. All right. Now, up here again, where would I find that? That's down here in the subcutaneous or hypodermis or superficial fascia. So down here, this will be adipose. Down here. Okay? So stratified squamous in the epidermis, areolar in the papillary region of the dermis. Skip this for now, and then down here we got adipose down in this subcutaneous layer. All right? Next up is reticular. Reticular connective tissue, no nice way to say this. When I look at it, looks like vomit. That's what it looks like. It looks like you weren't feeling too well, and you look at it, and when you look at the slide, you're going to look at it and go, yeah, it looks like vomit. Or think of it this way. You have a cat, it's 3 o'clock in the morning, you're dead asleep, underneath your bed, you hear, you're like, oh man, because you know they're going to hork it up down there. When you look at it, bada bit, it looks like reticular, okay? Keep in mind, when you look at reticular close up, you'll see these thick cords running through it. That would be the reticular fibers. You're not liable for that, but that's what it is. Reticular connective tissue, again, is found in squishy organs. Think of like the liver, the spleen, your lymph nodes. It makes them squishy. It holds them together. Those are the 3 types of loose connective. Then we have 3 types of dense connective. You have dense regular, dense irregular, and elastic connective. All right, so dense regular. Dense regular connective tissue, we're going to have collagen fibers. Doesn't matter how they're oriented, but this is important. The collagen fibers have to run parallel to one another. They don't cross each other. Where would you see this? Tendons, ligaments, and what are called aponeuroses. What's an aponeuroses? Take a tendon, make it really wide, like your scalp. That's an aponeurosis. Avascular, if you rip it, you can't fix it. But again, the collagen fibers run parallel. What you might see in the slide is every so often like a little dot here or there in it. Those are the fibroblasts. Now, dense irregular connective tissue. Dense irregular connective tissue has these large collagen fibers, but now it looks like spaghetti in a spaghetti bowl. All the fibers are going everywhere. Why would I leave my drawing up? Because that's going to be here. So what you would see are collagenous fibers like spaghetti noodles go in all different directions, all in here. This upper layer of the dermis is called the papillary region. This lower region down here, this is going to get a little confusing, is called the reticular region of the dermis. Now, not reticular fiber, not reticular tissue, reticular region of the dermis. That's the lower dermis. The reticular region of the dermis down here is going to have dense, irregular connective tissue, collagen going everywhere. When I mentioned before stretch marks, that's because of those, okay? That would be torn and then make parallel strips. All right. Now, moving on then, elastic connective tissue is going to have elastin in it. Elastic connective tissue is going to be around blood vessels. Think about the major vessels coming out of the heart, like for the artery, like the aorta. It's tissues that have to have some sort of structure, but then stretch, go back, like a major blood vessel. The one you have on canvas, if I remember right, is the aorta. Not the upper surface. Keep in mind that would be simple squamous. The area down below. When I look at it, how I think about it, it looks like a bloody sponge. Like you took a kitchen sponge and you soaked it in blood. That's what it kind of looks like to me. But that would be elastic connective. Next group. Now we talked about loose connective, dense connective. The third subgroup is now going to be cartilage. Therefore, what you would hear is then chondro. Okay. Cartilage, again, is going to be avascular, can't repair itself. There's going to be nerve endings when it's an adult. It's there for strength. So keep in mind, the chondroitin sulfate is kind of like a rubbery substance. Give you an example. You can do this with your nose, do this with your ear. That's chondroitin sulfate. It's kind of a rubbery consistency to it. So therefore, so in your cartilage tissue, you're going to have chondroitin sulfate. That's going to make it flexible. You are now going to also have collagen through it, which is also, he says, going to create strength with flexibility. All right. So here, very important subject. In cartilage, what you're going to have are little cells. So if I have a cell that is separated from everybody else, that is going to be called a chondrocyte. That chondrocyte is going to have a little bubble around it. That little bubble is called a lacunae. So the lacunae is the house, the bubble, the membrane that the chondrocyte lives within. Now, if it's by itself, it's called a chondrocyte. If when you look at it and the cells are like this, they're right next to each other and there's hardly any room between them, that means they're younger. Those cells would be chondroblasts. If they space out, because again, as they get older, they secrete more and more hyaluronic acid, and like islands, they drift apart from one another. Whenever they have space between them, they're an island unto themselves. That would make them chondrocytes. Okay, so first one you see there is hyaline cartilage. Hyaline cartilage is the most ubiquitous cartilage found in your body. Ubiquitous means found everywhere. Hyaline cartilage is going to be areas where you need support. Follow the tip of your nose, that's hyaline cartilage. The trachea, right here in your windpipe, to hold your windpipe open, that's going to be hyaline cartilage. When you were inside mommy developing, your embryonic axial skeleton, your arms, your legs, your fingers, all that was hyaline cartilage first. It laid a foundation, a template from within which your bones would form later. As you get older, you take a bone, think us through. Let's just say I have your femur. Okay? That was all cartilage when you were inside mommy as a fetus. Roughly about, we're looking at 6 to 8 weeks of age, this is going to start to ossify. Well, think. Where do you start to ossify? At the tips and ossify toward the middle or toward the tips? You always ossify from the middle and then work your way to the tips. Why? Because if you did it the other way, your bones would never grow any longer. Your growth plates, now understand, once this bone ossifies, you're going to have cartilage plates here and here. Those are going to be called then your epiphyses. It's the epiphyseal plates, I should say. So also called the metaphyses. But epiphyseal plate is your cartilage plates, your growth plates. They're made of hyaline cartilage. Eventually, as you get taller and taller, you don't get taller anymore. Why? Because the hyaline cartilage gets all used up and therefore doesn't divide anymore. The bone doesn't get longer anymore, and you maintain whatever height you are. So hyaline cartilage can be found in those plates as well too. Anywhere I missed important, just making sure. No, we're good. All right, so that's all hyaline cartilage. Now, hyaline cartilage, when you look at the pictures, note the size of the lacunae and chondrocytes. They're not teeny, they're not enormous. They're kind of middle of the road. Let's see. So, one picture I put on there for you is the fetal finger. What you got to imagine is you're looking at this part. You're looking at the metacarpal coming up and phalanx number 1 right there. That knuckle is what you're looking at. So where you see it's rounded, that's that right there, okay? When you look at it, you'll see like these little clear bubbles with a cell in it. The bubble is the lacuna, the cell is the chondrocyte. The blue shimmery stuff around it, that's going to be the chondroitin sulfate. When you look at the next one, that's going to be the growth plate of a bone. What you'll see is kind of like this plate coming down, and then there'll be this like moon shape of tissue, and then it finishes off. That right there is the hyaline cartilage. That's the growth plate. That would be the epiphyseal plate of a growing long bone. All right. Next, fibrocartilage. Fibrocartilage looks like you went to a county fair, and you know when you go to county fairs, there's different types of cotton candy. There's pink and blue. People will tell you they taste different. No, it's just spun sugars, your brain talking to you. But let's just say you're in a county fair, you got the blue cotton candy skipping around, county fair. But as you know, it's summertime, and therefore gnats are out, and the gnats get caught in your cotton candy. Congratulations, you now see fibrocartilage. So the fibrocartilage looks like blue cotton candy going everywhere, and then you see these little specks inside the fibers. Well, the blue cotton candy fibers are collagen, and then the little specks in it are chondrocytes. What is fibrocartilage used for? It's a shock absorber. Basically, it's to prevent bone from hitting bone. So your intervertebral discs, the cartilage between your vertebrae, so that when you walk, they don't smack your vertebrae together. The menisci in your knee, so that your femur and your tibia don't hit each other. That's all fibrocartilage. They're shock absorbers. How's it a shock absorber? Through the collagen, taking the pressure and dispersing it over a wider area. Elastic cartilage. Elastic cartilage, when you look at it, the lacuna and the chondrocytes are huge. Where do you see elastic cartilage? Real simple, right here. If you didn't have elastic cartilage, you took your pinna, which is your ear, and you folded it down. When I let it go, it wouldn't pop back up. That is elastic cartilage. So therefore, you see it there. You see it in your epiglottis. Your epiglottis is right above your windpipe. It's a little flap that covers it. So when you swallow, if you take your fingers and when you swallow, you'll feel your neck go up. What does that do? It makes your epiglottis flop over your windpipe. So whatever you're swallowing doesn't go into your air pipe. It goes behind it, down your esophagus, into your stomach. Alrighty, let's see. So, elastic cartilage again is going to give flexibility with shape. Now, last two categories. We got osseous bone and then blood. Then we still got muscle and nervous tissue. So, let's get through all this. Now, bone. Bone's simple. Think of it kind of like rings of a tree, okay? You've all seen a stump get caught down before, and therefore you have what looks like this. Alright? The middle hole in the middle. The middle hole is called the central canal or Herbacean canal. That's where blood's going to come through. These little rings coming out from them, these are called lamellae. What are they? The lamellae are growth rings. In the lamellae, what you would see are these little black specks that look like raisins in them. What are the raisins? Those are the lacunae. Inside of them would be the osteocyte. You can't see the osteocyte because this bone is too dense and the light won't pass through, so you can't see it. But you got to know the little speck is a lacunae. Inside of it would be the osteocyte. All right? Now, so we have the central canal, also known as Herbacean canal, the lamellae. Well, how the hell then do I get the blood from here all the way out to here? When you look at it, you'll see these little fine cracks that radiate out from the center. These little fine cracks are called caniculi. Think like Venice. Canals, little tiny canals, and therefore the blood is allowed to come out to nourish the cells, and their waste can get pumped out. Okay? This whole structure is called one bone unit, and that is called an osteon. One bone structure. This is specifically for compact bone. Compact bone is what you think of when you look at a skeleton and it's all nice, smooth, white. Compact bone is also found in the shafts of your long bones. So when you think of a femur, when you think of your radius and your ulna, that's all compact bone. It acts like a lever, like a seesaw, in order for the muscle to work on to cause a desired motion. All right, let's see. And everything I labeled, you're liable for, just so we know. That would be bone. Blood, if you remember, doesn't connect anything, but it derives from connective tissue. Blood, again, the ground substance is plasma, and then what you would see are a bunch of little balls floating around in this. The area between would be the plasma, the ground substance. Little balls that don't have a nucleus, those are erythrocytes. Erythrocytes are red blood cells. You have to be able to label them as erythrocytes. Erythro is Latin for red cell. Now, what do? They carry around gases. They carry around oxygen and carbon dioxide. So it's for gas exchange. Every so often, somewhere in here, you would get a larger cell, and that larger cell would either have a nucleus, or it might have a multi-lobe nucleus. So basically, if it has any nucleus at all, that would be a leukocyte. Leukocytes are your white blood cells. Difference between them, like basophil, eosinophil, all that would be more when you go to AP2, or if you ever run across me in immunology again, then we go over that as well. But for now, all you gotta know is that's blood, it's connective tissue, that's an erythrocyte, those are leukocytes. All right. Then you also have your platelets in there, which are thrombocytes, which then clot your blood. Assuming you're not a hemophiliac, you don't have to label those. Now, for that then, I'm gonna get rid of this. And then we come to our last two major categories, muscle and nervous. All right, so muscle. Muscle, if you remember, and nervous are both excitable tissues. They both need some sort of stimulus in order for them to work. There are three types of muscle tissue: skeletal, cardiac, and smooth. You gotta be able to differentiate between the three. The categories we look at is, number one, are they striated, meaning are they banded or not banded? Are they voluntary or involuntary? Do they fatigue or not? Fatigable, non-fatigable? Are they uninucleated or multinucleated? That's how you have to compare and contrast between these, okay? So first off, skeletal muscle. Skeletal muscle, when you look at it, looks like bacon because it is bacon. All right, so with these, you're going to have long protein fibered cells run through an area. If they run parallel to what you're looking at, you will see areas where it's like a light pink, dark pink, light pink, dark pink. Those are striations. So skeletal muscle is striated, banded. God, I hope it's voluntary. Otherwise, you'd be, twitching all over the place, okay? Voluntary. They are fatigable. If I had you take a 1 pound weight and hold it at your shoulder, eventually it would fall down. They do tire. Last but not least, big, huge thing, they are multinucleated. So if you see cells which are multinucleated with bands, that's skeletal. Why are they multinucleated? That just means that many, many, many smaller cells fuse to make one long cell. Keep in mind, some of the largest cells in your body are 30 centimeters long. These are individual muscle cells that come from your hip all the way down to your knee in a muscle called your sartorius. Those are single muscle cells. Well, how the hell do you make it that long? Because you fuse many cells, and therefore the nuclei are kept. All right, cardiac muscle. Cardiac muscle are going to be shorter cells. They are only uninucleated. They are involuntary, otherwise you'd just sit here and go, beat, and you forget, you'd go into cardiac arrest. Along with that, I hope non-fatigable as well. They don't tire. Like skeletal muscle, they are striated, they're banded. When you see cardiac muscle, it kind of looks to me, whenever you see it stained, it looks like red bamboo. So you'll see kind of like these strands, and they'll be stripped like this. Those are the striations. The important thing about cardiac muscle tissue is that where the nuclei are, there's going to be this little tiny thin line in between adjacent cells. That is super, super, super important. That little line right there is called an intercalated disc. That intercalated disc is what allows the nerve impulse to jump from cell to cell to cell. It's the conduction wire. Think about it. When your heart beats, it doesn't go boom. It goes ba-bump. Why? Because a wave has to go from one side all the way to the other side. To do that, you have to have a connecting wire. The intercalated discs allow that to happen. Be careful when you answer questions about where cardiac muscle is found. Don't just simply say the heart. Because remember, the heart has an outside surface and an inside surface, and the wall. The wall is the cardiac muscle, plus the major arteries leaving walls of the cardiac. Smooth muscle. Smooth muscle tissue, again, is going to be uninucleated. It's involuntary. It's non-fatigable, and this is the only one that's not striated. Some smooth muscle looks like taffy that you take and you keep pulling to where it almost breaks. Long, spindly cells with one nucleus. So when they're teased apart, smooth muscle cells look like these long, skinny cells with a nucleus in it. It looks like somebody stretched and they're almost going to break. So when you see it on canvas, you're going to have ones which are teased apart. These are these individual ones, like little threads with one nucleus. Whenever you see them in the tissue, specifically the pictures I gave you for the small intestines, you'll see like this pipe that looks like this, and then you'll see an area up here that'll kind of look like this. Well, from before, these cells, if you look at them, those are the simple columnar cells with the goblets, simple columnar with goblets, and that would be how you're absorbing things into your bloodstream. But then you'll see wrapped around it, almost like insulation, these cells, which are long, spindly cells going in a circle. Well, that's the smooth muscle. What does that do? Like I said, your small intestines is like a tube of toothpaste. They have to contract in a rhythm to move the contents throughout your small intestines. So therefore, that's what allows that to happen. That's smooth muscle. All right. Last but not least, then we have nervous tissue. Nervous tissue, as you know, is for communication, as well as for coordination of body activities. It also allows you to pick up a stimulus and hopefully elicit the proper response, and it's also excitable. For this, you're going to have a neuron. Neurons have a large central area with a nucleus. This is called the nerve body, or also known as the soma. On one side, it's going to have what look like tentacles coming out one side and a cord coming out the other. These tentacles coming out are called dendrites. The one cord coming out is called the axon. Dendrites are what receive the nerve impulse. The soma has the nucleus, interprets it, and then sends the nerve impulse away along the axon. Surrounding, now this whole picture is a neuron for nervous tissue. Surrounding it, you will see these little tiny cells surrounding the neuron. These little tiny cells are called neuroglial cells, sometimes just called glial cells. What are they? They're kind of like the nursemaids that take care of the neuron. They're there to nourish it, take care of it, remove waste, okay? Because the neuron has its own things it's got to worry about, more or less. Now, all you have to know for your test is when you see something like this, when you see basically it looks like a paint splotch, that's nervous tissue, and that's called a neuron. All the other things I'm pointing out to you would be more or less when you go to AP2, when you start going into the nervous system. I think that's everything for histology, okay? So next lecture will be on skin, integumentary. Thank you.